Hello, and welcome to this afternoon's MDF Meet the DM Drug Developer webinar. I am Mike Knoppen, the MDF Program Director. Today, we are very honored to have with us our partners, Arthex Biotech, to talk about their drug development pipeline for myotonic dystrophy. Here's a little information about the foundation. Our vision is a world with treatments and a cure for myotonic dystrophy. Our mission is community, care, and a cure. And by that, we mean that we support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research toward treatments and a cure. The Myotonic Dystrophy Foundation is pleased to make available for free many resources for our community members, including some you see on your screen now. Our toolkits and publications are available for download at myotonic.org/toolkits-publications. There you'll find medical guidelines to share with providers, toolkits to teach you and your family more about myotonic dystrophy, and many other important resources related to school, social security, and many other important topics. Support is crucial in our community, which is why we have many support programs available for the whole population as well as specific interest groups. You can find all of those at myotonic.org/find-support. To find out what's going on at the Myotonic Dystrophy Foundation, check our calendar of events. At myotonic.org/calendar/month, you'll see everything that's going on this month, including support groups, webinars like these, and many other important and fun events. The Digital Academy of the MDF holds all of our video resources, such as recordings of webinars like these. You can find them at myotonic.org/digital-academy. In addition to today's amazing Meet the DM Drug Developer session, we have a few more scheduled this year. If you join us on July 1st, August 5th, you will hear from our partners Avidity Biosciences and PepGen about their drug development pipeline for DM. And if you join us in September 9 and 10 at the MDF Annual Conference, you'll get to connect with a number of companies working on drug development in myotonic dystrophy and hear several industry updates. To register and submit questions for upcoming Meet the DM Drug Developer events, please go to myotonic.org/meet-dm-drug-developers. In addition to that webinar series, we have an Ask the Expert series where folks can learn about a number of topics important to the community. Upcoming events include a community portrait through surveys and insurance claims data, where our friends at the Marigold Foundation will present on a number of research findings over the years that paint a picture and tell a story about the DM community. In July, you'll hear from a palliative care neuromuscular expert. And in August, you'll hear from a disability rights lawyer about future planning and special needs, including information about special needs trusts. And again, you can get a lot more information and ask your questions of experts at our MDF Annual Conference in San Diego on September 9th and 10th. To register and submit questions in advance for any of these Ask the Expert sessions, please go to myotonic.org/ask-expert-series. Now, it's my great privilege to introduce our two speakers for today's Meet the DM Drug Developer. The first will be Dr. Beatriz Llamusi Troisi. Dr. Llamusi received her master's degree and PhD in biochemistry from the University Miguel Hernández in Spain. During her PhD, she focused on the molecular bases of neuronal regeneration. After that, she began working in the Department of Genetics as a postdoctoral researcher, dedicating more than 13 years to the research of pathogenesis mechanisms and therapeutic approaches in rare neuromuscular diseases. During this period, she produced 22 scientific articles and 7 related patents. She was PI in 5 competitive projects and supervised 4 PhD projects. During the initial steps of the project that led to Arthex Biotech's founding, she was awarded several valorization grants, including training in business management development, IPP, financials, and regulatory issues. She co-founded Arthex Biotech in December 2019 and has served as CEO since that time. Our other speaker today will be Dr. Judy Walker. Dr. Walker started her career as a clinical neurologist and clinical trial investigator in Canada. In the late 1990s, Dr. Walker joined industry and has spent the last 25 years in a variety of clinical and medical leadership roles in the US, Switzerland, UK, and Singapore. Her focus throughout that time has been to bring effective and safe drugs to market for patients with some of the most disabling neurological conditions, including many rare diseases. At Arthex, Dr. Walker is responsible for all clinical and medical activities in the company, moving Arthex's lead investigational compound, ATX-01, into clinical studies for myotonic dystrophy, preparing for ongoing development of the drug pipeline, and building a highly skilled team to ensure success. She's excited and humbled by the potential of Arthex's investigational drug, ATX-01, to have a meaningful impact on DM patients' lives. We invite you and encourage you and appreciate you in supporting the MDF mission. If today's program speaks to you, please consider showing your appreciation with a donation to MDF. Together, we are changing the future of myotonic dystrophy. You can make donations at myotonic.org/donate. And now it is my great pleasure to welcome Dr. Llamusi and Dr. Walker.
Hello, Michael. Hi, thank you very much. You hear me well, I guess, right?
Yes.
Okay, thank you for the nice introduction and thank you all for being there today. It's a great opportunity that we have today for sharing our project at Arthex with you. As Michael mentioned, we created the company at the end of 2019 and our first product in the company was ATX-01. An anti-MIR-based drug to treat myotonic dystrophy disease. This is the disclaimer because I might be doing some forward-looking statements. And well, this is where I am right now at the Park Scientific at the University of Valencia, as we are a spin-off of the university. And Arthex's mission is to develop treatments against for unmet medical needs. So diseases that have no treatment. And the way of doing it, it's that we have selected, it's by controlling the levels of microRNAs to modify gene expression. So you have probably heard a lot about genes and proteins, but maybe microRNAs is not so, it's a kind of molecule that is not very well known by you. But just to let you know, microRNAs are small RNAs, small molecules, which are naturally synthesized by the body in order to modulate the level of expression of hundreds of different proteins. So we believe that they are very good therapeutic tools because they are easy to target in a very specific way. And just by doing small modifications in one microRNA, we can have a very big impact in a whole pathway. So we can modify different proteins just by touching one microRNA. So as I was mentioning, the focus of our company right now, we started as a one-product company focused on myotonic dystrophy. Here we are developing a product that is called ATX-01 against a microRNA that is called MIR23B. So with this ATX-01, what we are doing is to block the expression of MIR23B. And our estimation is that we will be submitting the IND by the end of this year. So hopefully in the beginning of next year, we will be already in the clinical trial. Why we focus so much on myotonic dystrophy is just because of the strength of myotonic dystrophy community. So just because of you, all of you interested in this disease in some way. Here I use this photo of Antonio Alvarez with his wife. It's in fact his WhatsApp profile, because he is the person coordinating BENE association in Spain. We were in a very close contact from the very beginning, already in the university, and his wife was always asking us, when are you going to do the pill that will be helpful for my children and also for my husband? Well, finally, we created a company because of them, because we really want maybe not a pill, but maybe an injection that could be helpful. So, well, you already know better than myself what is myotonic dystrophy. So just to remind you, it is considered a rare neuromuscular disease because of the prevalence, that it's not very high, but still there are almost 1 million patients all over the world of this disease. Only between US and Europe, more than 100,000 patients. So it's a very important disease that until now it has no treatment, and it's mainly affecting adults and also pediatric population. And you already have heard a lot about the mechanism of action of this disease. You know that it's quite complicated when we all try to explain, but the good thing is that it has been very well described. So all the molecular mechanisms behind the disease is very well understood by the scientists. So just to remind you, the genetic cause of the disease is this expansion inside the gene DMPK. So there is a region inside this gene that is expanded, so it's longer than in the healthy condition, especially when this region, when this triplet of CUGs is expanded and there is more than 50 CTG repeats, then the symptoms start. And what it has been very well described is that with longer CUG repeats, the symptoms of the disease are more serious. However, the symptoms are not related to this gene DMPK. The symptoms have been related to a protein which is called MBNL. In fact, MBNL is a protein that is regulating the expression, the alternative splicing of other proteins. Specifically, some of these other proteins regulated by MBNL have been shown to have an important impact on the symptoms of the patients. For example, this is a very clear example, the insulin resistance. The insulin receptor, it's affected in the disease, causing the insulin resistance in the patient. And this protein is directly regulated by MBNL. So the main symptoms in the patients have been linked with the loss of function of this MBNL. And the molecular effects that are linking these two factors can be explained here, because this expansion that is present inside the DMPK is producing what is called a toxic RNA. This is considered toxic just because it has a deleterious effect. So this extra long RNA, it's folding into a hairpin. It has a secondary structure that is acting as a sponge of MBNL proteins. So these proteins have affinity for this RNA, and they get sequestered here, causing what we call ribonuclear foci inside the nuclei. So you can see that these are muscular cells from patients, and these circles are the nuclei of the cells, and you can see these dots in green, which are the nuclei foci. These are the MBNL accumulating inside the nuclei because they get trapped here. So this sequestration of MBNL is causing part of the loss of function. But we have also established that one of the effects that of this toxic RNA is to produce an overexpression of one microRNA called MIR23B. So in the patient muscles, we have observed that this molecule, this regulator RNA, it's overexpressed. And one of the functions of this molecule is to repress the production of MBNL protein. So you can see here that the final loss of function of MBNL is caused by these two different mechanisms: the sequestration in the hairpin and also the repression exerted by this MIR23B. As you previously heard from other companies, most of other approaches are focused, therapeutic approaches are focused here. They are trying to destroy this toxic RNA or to inhibit the interaction between the protein and the RNA. But in Arthex, we are focused on MIR23B. And this is because we believe that this microRNA is a very good therapeutic target for myotonic dystrophy. Of course, it's something novel, and it's also always good to have different therapeutic approaches in a disease. But MIR23B is especially good because it is very well conserved. This means that this molecule is exactly the same in all these species. You have here humans, mice, rats, mini pigs, dogs, non-human primates. And you can see that the sequence of this microRNA, it's exactly the same from one species to another. That means that it's a very important microRNA, and also that if we can block this microRNA in mice, we will also do it exactly the same, with exactly the same efficacy also in humans. So we can use the same molecule in the mice models of the disease, and also in the humans. In addition, MIR23B is important because, as I mentioned, it is upregulated in the muscles of the patients. So this is part of the pathology. You can see here in this graph, we show the levels of the MIR23B in control or healthy patients in white, or in DM1 muscles, which will be in gray. You can see that they are MIR23B is higher in the patients of the muscles, but also here in mice, this is gastrocnemius muscles, quadricep muscles of a mice model of the disease. It is also upregulated in the model, DM1. These are patient-derived myoblasts, so cells from the muscles of the patient. And you can see that MIR23B is also upregulated. And especially you can see that in cells these cells are coming from patients with expressing different lengths of repeats. So here the patient expressing 1,300 CTU repeats have more severe symptoms than a patient expressing 300 CTU repeats. And as you can see, the levels of the MIR23B is higher in patients with longer repeats. That means that the levels of MIR23B correlate with the severity of the symptoms. That makes it especially relevant for the disease. In addition, it is targeting directly MBNL, which is the main protein linked with the symptoms. And there is already a KO mice where the scientists have depleted completely the expression of this MIR23B, and they have any phenotype. They have found any phenotype, meaning that the reduction of this MIR23B will be safe also in humans. So looking for the most efficacious way of inhibiting MIR23B in order to obtain an upregulation of MBNL, so what we wanted to do is to block this inhibitor to produce an overexpression of MBNL. So to do it, we have designed anti-microRNAs. That means an oligo sequence that is complementary to the sequence of MIR23B. So this is a single-stranded RNA molecule that can bind to MIR23B and simply block it. So we have designed a very, very specific oligo that can bind to MIR23B to block its activity. And we have used a combination of chemical modifications in this RNA that allow this RNA to be very stable. And also to have, in order to have an endurable therapeutic effect. In addition, some of the chemical modifications we have introduced is protecting this RNA to be in order to avoid detection by immune system activation. And we have tested the effect of a group of these oligos in screenings in vitro and also in vivo in models of the disease, looking for the best oligo and the safest oligo to inhibit MIR23B. In addition, what we wanted to do is to inhibit MIR23B, but only in those places where are affected by the disease. So, as you know, although myotonic dystrophy is known to be mainly neuromuscular, and there are a lot of systems affected, but you can see that the main symptoms in the patients, or the more severe symptoms, are affecting skeletal muscle, respiratory system, also cardiovascular system, and cognitive function. Especially cognitive function is more affected in the pediatric or in the congenital population, but also in adult DM1 patients. It will be very important to be able of treating nervous system. So what we have done is to use hydrophobic conjugates that we can link to this anti-microRNA in order to bring the molecule to the places where it is needed. This molecule, what it's doing is to help us to increase the concentration of our molecule in the target tissues for this disease, which will be mainly skeletal muscle, cardiac muscle, and nervous system. But also it's allowing us to obtain less concentration of our molecule in other tissues, such as the kidney or the liver, as we don't want to target them. So we also perform a screening looking for the most efficacious conjugate to help us or to allow a tissue-focused delivery of our molecules. Finally, putting together these two components, we created this enhanced anti-MIR delivery platform with the specificity of the anti-MIR oligo and the tissue enrichment that will provide the hydrophobic conjugate. We created the molecule that we call ATX01. This molecule has a higher effect in muscles, cardiac, including cardiac muscle, and also brain. And especially in the muscles, we have increased the potency by 10-fold. And it's endurable. It's not producing an activation of the immune system. It has no hemolytic potential, and it's safe. It is safe for the kidney and the liver. The effect of ATX01 is the following that you can see in this graph. This molecule is binding to MIR23B, so it is blocking this microRNA. Then this microRNA can no longer repress MBNL translation. So finally, we have an upregulation of this protein. You can see these results that we obtain in vitro in a human derived myoblast. This will be myoblast or muscle cells from controls, muscle cells from DM1 patients, and this will be DM1 cell patients treated with our molecule. As you can see, we achieve around 4-fold more higher levels of MBNL protein in human cells. We have also tested this molecule in a mouse model of the disease. This mouse is the HSALR mice that is it's commonly used as a very good model for skeletal symptomatology in myotonic dystrophy. And as you can see, when we were giving here a single dose intravenous administration of our drug at 24 mg/kg. And as you can see, we have higher level of the protein in quadriceps, gastrocnemius, so skeletal muscles, also in diaphragm, which will be very helpful for the respiratory problems of the patients. But importantly, in heart and brain, which are two tissues that are very difficult to target with oligos. In fact, most of the therapies right now in development to treat myotonic dystrophy, they are able to target skeletal muscle and also cardiac muscle, but not the brain. And we see here target engagement also in the brain, but not in other tissues such as the kidney, or reduced upregulation in other such as the liver. As I mentioned, we are able to increase MBNL level in human cells, but what is very important is that we can also do it in, as I mentioned, that this microRNA is very well conserved through species. We have shown that we can also upregulate MBNL in rat cells and mini pig cells. So these will be the two species that we will be using to test in the toxicology package for the preclinical development of our drug. And this has been already accepted by the FDA, that as the molecule and also the mechanism of action is so well conserved, we can use these two species for the preclinical development. This is the mechanism that I have shown for ATX01, this upregulation, this direct upregulation of MBNL, but we have also discovered that in human cells, ATX01 has a second very important effect. This is what we call a dual target therapeutic approach, because reducing the levels of MIR23B, it's also producing a downregulation of DMPK. You can see here that with less available levels of MIR23B, we are reducing the levels of DMPK. So here in the nuclear cells, you do not see the foci anymore. You see now MBNL spread in all the nuclei, how is the original distribution of this protein. So what we have done is to quantify the number of foci in the DM1 cells treated with ATX01, and as you can see, we are decreasing importantly the number of these foci. And this is important because DMPK reduction is what other companies are using as a measurement of their target engagement. Other companies that are using ASOs, for example, against DMPK, are always focused on reducing DMPK foci or DMPK expression. And this is, so DMPK, it's already a validated target in DM. And this is exactly what we are doing also by targeting MIR23B. So we have two activities with the same molecule, which is the main advantage of working with microRNAs, as I mentioned at the beginning. We can have a direct upregulation of the protein, but in addition, we are reducing DMPK expression. So finally, two mechanisms that are producing more available MBNL to perform the rescue of the splicing and the rescue of the symptoms in the patients. We have done a lot of efficacy studies using ATX01 in different models of the disease. I just show you here the results we obtained with different lines of human primary myoblasts. What we wanted to do is to test ATX01 in as many different lines from patients as possible to see that ATX01 could have effect in very different genetic profiles. So even in patients with very long repeats, we could have an effect. As you can see, for example, here in these cells, you can see in blue the nuclei of the cells. In red, these dots are the toxic RNA. And when we treat with ATX01, we are reducing importantly the toxic RNA. In addition, you can see that if we labeled MBNL with green, the normal distribution in a control condition will be this is dispersed in the nuclei, but in DM1 it is importantly downregulated and it's located in these foci, in these dots inside the nuclei. Now, with the treatment of ATX01, it's again dispersed in the nuclei. In these cells, we can quantify several molecular effects of our drug, such as this reduction on MIR23B expression when we treat with ATX01, overexpression of MBNL protein and reduction on DMPK. In addition, we can also measure some myoblast differentiation markers because our molecule is improving the differentiation of these cells. As you can see, in DM1 cells in comparison to controls, the fusion index and myotube area of the cells are reduced because they are not differentiating well, and when we treat them, we are increasing both values. In addition, we can rescue splicings. You can see that some of the splicing regulated by MBNL are here repaired by ATX01. So we have percentage of rescues of these splicings around 50 up to 60%, which means that the MBNL that we are upregulating, it's functional. And the rescue of these splicings is very relevant because you have seen probably in many talks that there is a correlation between the splicing and the strength in the patients. In fact, it has been published that only 20% improvement in the splicings could be the difference of 1 grade in the ankle dorsiflexion strength test, which could be maybe the difference between walking and non-walking for a patient. So very small splicing rescues could have a real very important functional correlate. What we have done here is to measure in human DM1 cells, you can see in this graph, how with higher levels of the protein MBNL, we have higher level of rescue of PSR that we call, this is the percentage of rescue of the splicings. Here, just with twofold upregulation of MBNL, we are able to rescue at least 30% of these splicing. So we do not need a very big upregulation of the protein to obtain an effect. And the splicings are seen as the rescue of splicing is a potential surrogate biomarker of efficacy in a future clinical trial that we will like to assess. This is the one of the studies that have been previously published where they were showing this correlation between the strength of ankle dorsiflexion here and the levels of MBNL or the splicing rescues. What we have done in cells is to study all this group of splicings that have been shown to correlate especially well with the strength in the patients to see if our treatment was able to modify and to rescue these splicings here in brown. What you can see, all these dots mean that there is a difference between control condition and DM1 cells, so cells from patients of DM1. The bigger the dot mean bigger difference between controls and DM1. As you can see, when we treat DM1 cells with ATX01, most of these dots disappear just because there is no longer difference between control and DM1 cells. And we have quantified that we are able to rescue around 85% of these splicings, which are functionally relevant. So hopefully we will try to see this in the patients in the future. We have some proof of concept also done in a mouse model, which is called HSALR, which is an adult onset model, because these mice are expressing around 220 CTG repeats, which is the average in the adult onset patients. What we were doing here is to give one intravenous injection to these mice, one every 30 days, so one monthly injection, 3 injections in total, and at 2 different concentrations. Even with the lowest concentrations, with just 3 mg per kg, as you can see, the strength of these mice, the force, was almost completely rescued. The difference between the lower dose and the highest dose is that with the dose of 24, we have a more durable effects. What we are measuring in the muscles of these flies, as a measurement of these mice, as a measurement of the target engagement, the measurement of the molecular effects of our therapy, it's simply the levels of the MBNL protein. So we can see that in the mice, we have a dose-dependent upregulation of MBNL protein. And as a result, we also observed a rescue in the splicings of between 20 and 50% of the splicing in these mice. And something important is that using these mice, we are underestimating the potency of our therapy because these mice are not expressing the human DMPK with the repeats. So we only see the effect of our therapy on the upregulation of the protein, but not on the DMPK levels. That this is something that we will be able to see when we are treating humans. Apart from the efficacy studies, we have done a lot of toxicology studies in vitro and of course in vivo. In vivo, we have done studies already in rats, dose range finding in rats, maximum tolerated dose in mini pigs, and in non-human primates also, we have tested immunotoxicity. And we are just initiated the GLP studies in rats and mini pigs that will enable our studies in humans. Until now, the results have been quite positive. And just to let you know, well, we have not even reached toxicity in mini pigs or non-human primates, even using doses that were about 30 times higher than the efficacious dose. So that means that the therapy is quite safe. But something important related to this is that we can measure the levels of the protein in all these animals. So we have seen that in the rats, for example, we see an upregulation of the protein when we treated the rats. So we are giving here 3 intravenous injections of ATX01 at 8 and 30 mg per kg, and we were already in a very high level of protein. Even here you can compare between one intravenous injection or subcutaneous injection. So we have seen that also with subcutaneous we could have an effect. And this is the levels of the protein in a time point study. So after one single injection, we sacrifice the animals at different time points. And we have measured the levels of the protein. And we have seen that even 22 days after the injection, we have around 3-fold of protein, which will be more than enough to see a rescue of splicing. So we could space out the doses enough to give a comfortable treatment to the patients. Just to show you also in the mini pigs studies, we have seen a dose response. So we have seen target engagement, and we can measure MBNL also in the muscles of these mini pigs. So we are sure that we are giving doses that are therapeutic and safe, both. So just the brief about all these preliminary studies, as I have shown you, we can measure MBNL levels as a measurement of the target engagement in the preclinical animals, and we will also do it in the patients in the future. We have shown that we have sustained efficacy, and we could space out the doses at least to give once monthly dosing in the patient. As I mentioned, the therapy is quite safe because we have seen that we have a wide enough therapeutic window, and as mentioned, we have not even reached toxicity in the mini pigs or non-human primates. And of course, we still have to work a lot with the results on the non-human primates, but the results we have until now are very positive. It's safe and it's not activating complement. So we are still ongoing with other experiments in the animals, but I will pass you now the stage to Judy so she can give you more details about our clinical trial plans. Please, Judy.
Great, thank you. Thank you so much, Beatriz. So following on from what Beatriz said, you know, we are very much preparing to go into a clinical trial. Many of you asked, when are we going to go into a clinical trial? Where we're in the preparation stage, and we are hoping to be dosing patients early next year. We are completing our clinical toxicology package. So before the FDA or the European regulatory agencies will let us go into humans, we have to complete some studies. But so far, everything is looking really great, both in terms of the changes we're able to produce in these animals, as Beatriz has shown you, that we think will correlate with clinical efficacy, as well as with the safety that we've seen so far. So all that is great. We had a meeting, as Beatriz mentioned, with the FDA last autumn, and we had a couple of the usefulness of that meeting was that it helped us with our planning. So first of all, as Beatriz said, they confirmed that the preclinical package that we were putting together was appropriate. And secondly, and importantly, from my perspective, who'll be running the clinical studies, hopefully with many of you that are on the phone, is that we are able to do our first clinical study in patients. So sometimes when drugs are developed, you have to go into healthy volunteers first. But in this condition, because of the type of product we're dealing with and the fact that there is this tremendous need, there is pressure to get drugs to patients quickly. So we are going to be doing our first in human study in patients with DM1. Now, this study is, and I'm going to talk a little bit about it, it's a somewhat of a complex study, but it's very similar to what you'll see some other companies doing. In other words, we're doing what's called an 3-part study that includes a single ascending dose, part 1, a multiple ascending dose, part 2, and an open label extension as part 3. Now, the interesting thing is that we're planning to study up to 5 doses. We may go higher, we may do fewer. We're going to find out how much we can give, but our main goal is to make sure that we are safe. So we've seen the safety in animals, but we're not taking for granted that when we do these studies, we do them in human subjects, and we have to make sure the compound is safe. So we're starting with the dose that is based on a dose that shows no effect in animal, and we're going to increase the dose, and we're going to do this in a very stepwise manner. And we're going to start by giving each patient will get a single dose. And the way we're doing this is that in each dose group, or each what we call cohort, there will be 8 patients. And out of those 8 patients, 6 will get the active treatment, 2 will get placebo. It'll be double blind, so we won't know who's getting what. The doctor, the nurse, the physio won't know, and the patient won't know. So this is what we call randomized 3-to-1. And then if that's okay at the first dose level, we'll move on to the next dose level. And if that's okay, we'll move on to the next dose level. At some point, we're going to start giving patients multiple doses. And this is where we move. And it's going to be based on safety. We're going to have a safety committee that's going to look at the results as we move forward and decide it's time to move to the multiple ascending dose part of the study. And then we'll start giving multiple doses. And we're first going to give 3 doses. Now, how far apart are we going to give these doses? Well, it could be every 2 weeks, it could be every month, it could be longer. How frequently we dose will depend on the results of the single ascending dose part, because we're going to look in this part not only at safety, but we're also going to look at the effects in muscles with muscle biopsies. And just like Beatriz showed you in the animal, we can see the effect lasting, how long the effect lasts in muscles. We are going to check that in human muscle to see how long the effect lasts in terms of how long do we see that suppression of miR-23B? How long do we see that elevation of the MBNL protein, and how long do we see, how much and how long do we see of rescue of splicing abnormalities? So that's going to dictate how frequently we dose, but we're going to give probably 3 doses in the multiple ascending dose study. And if everything's good, and if patients want to continue, then we will offer them an open label extension, probably for about a year. And so that's the overall design of this study. Now, the question is going to be, well, can you go to the next slide, Beatriz, please? And this is one of the questions that has come up, is who is going to be allowed to enter the study? Well, in every study, there's always what we call inclusion-exclusion criteria. And this is done so that we can make sure we have a relatively homogeneous group of patients to study. Because sometimes if you have too many patients that are too different, you don't see the results you want because some patients need a different dose. So what you always want to do is you want to start with a relatively homogeneous group of patients for your first study at least. And then as you move through development into later stages, you start broadening the group. So what we've decided to do is we're going to look first at adults with DM1 who are ambulatory, meaning they can walk. So our key inclusion are here, and I will say they're draft. And they're draft because we are still working on the study design. And we are going to be, one of the things that we're doing is we've spoken to a lot of experts in the field, and we've spoken to patient groups, and we've spoken to people who have developed these drugs before. But we're also going to be speaking a lot more to persons like yourselves, patients, and we're going to be doing some patient advisory boards so that we can make sure that we are designing the study appropriately. But at this point in time, what we're saying is we're going to look at patients who will obviously give informed consent, patients who have a diagnosis of DM1, and we're going with classic adult DM1 with more than 150 repeats in the DMPK gene in their white blood cells. You can be male or female, age 18 years or over, able to walk, and we are looking at patients who have myotonia. So we want to see, because that will help us see, do we have an effect on myotonia? Now, there's certain things that will exclude patients from this first study, and we want to have a group of patients that are relatively safe, because this is the first time we're going into patients. We want to make sure that there isn't any other confounding illness or other medication that could interfere with this. So we want patients who are safe and don't have any other major illnesses, in particular patients who don't have a really bad cardiac problem, because we don't think there's any problem with our drug in the heart. It's not that we have a concern about it, but we want to make sure that we have we don't have any safety problems. We are also going to ask that patients don't take mexiletine or other drugs that are used to treat myotonia. So patients who are on mexiletine, they can come off of it to come into the study, because we won't know if we're having an effect on myotonia if they're on the mexiletine. We want to make sure patients don't have hypothyroidism. And because we're going to be doing muscle biopsies, we want to make sure that we enroll patients who can have a muscle biopsy. In other words, they don't have a bleeding disorder, they're not on blood thinners. And we want patients who've not been on another investigational drug in the previous 3 months, because then it's hard to tell if there is an effect, whether it is due to our drug or to the previous drug and just a delayed effect. So this is where we are now. And as I say, things may change a little bit. So if you see something different, that may be why. We will, of course, have all our full inclusion-exclusion criteria on clinicaltrials.gov. And one of the questions we had is, how can I enroll in this study? Well, one of the best places to start is actually the MDF. So the Myotonic Dystrophy Foundation is fantastic, and they will be posting information on our study and giving you information on that. You can also go to clinicaltrials.gov. And I'll tell you right now that we are planning on working with the Myotonic Dystrophy Clinical Research Network. Now, we don't know yet which sites amongst that network, but these are some of the top sites in the US and Europe. And the network is expanding. So even if the doctor that you see isn't in that network, they could probably either refer you to someone in that network, or you never know, there might be a new site opening up, because we're in close contact with them and they are opening up more sites. So if you go to the next slide, the next question is, okay, so we know what patients we're going to be enrolling. We know what we're doing. So what are we going to measure? And one of the things we're going to measure, as I said earlier, this is first to make sure, any first study, you want to make sure, okay, this is safe. So our first goal always is safety. And we're going to do this by looking at what patients tell us. If they tell us this drug is making me nauseated, or if this drug is doing this, then we'll make note of that and we'll see what we can do about it. So safety and tolerability is the first thing. We're going to do blood tests mainly. We're going to do lab tests to make sure that we're not causing any changes in the labs. The next thing we're going to do is what we call, as Beatriz said, just like we did in the animal, looking at target engagement. In humans, we're going to do the muscle biopsies, and we're going to look at the same things that we looked at in all those different animal species. So we're going to look at the miR23B levels in the muscles. We're going to look at the MBNL protein levels. We're going to look at the splicing index. We're also going to look at the level of the drug we're getting into the muscle and the blood and the urine. Now, we expect, as we've designed this product, through a very, very, very robust process, so that it is not only specific, but that, as Beatriz says, it enters the tissues we want it to enter. So we're actually going to measure that in the muscle, because that's key. We have to get it to where it needs to be in enough quantity for it to have an effect. And of course, we're going to measure some what we call clinical endpoints. So these are things that might matter to you, that you can see or feel or hear. And now, those are always more difficult in a small study to get clear results on. So while an individual patient may see a benefit, sometimes when you add it all up, it's difficult to see. But we're still going to measure it so that when we go on to do bigger and longer studies, we know how to do those. Now, there's a lot of different what we call endpoints here. And I'm just going to talk a little bit about them. Some of you may already be involved in the END-DM1 natural history study that is being run by the DMCRN, the research network. And what we've done is we've mirrored the endpoints from that study. So those of you who are involved will be very familiar with many of these tests. So we have a lot of tests that are related to muscle, whether it's skeletal muscle, so you're running, you're walking, you're standing up, but also your respiratory function, your grip strength. We're going to look at myotonia. We're going to look at your tongue strength. So a number of different muscle testing that we're going to do. But because our agent, as Beatriz pointed out, also enters the brain and other tissues potentially, we're going to look at some other things. So we're going to look at cognition, and we're going to look at something called a cog state battery. Again, I think those of you in the END-DM1 study will be familiar with that. We're going to look at the eyes to see if there's any changes in the eyes. We're going to look at cardiac function, and we're going to look at metabolic status. We're going to look at, is there any change in the blood sugar and in other hormones. And we're going to look at some quality of life measures. Now, again, these endpoints, as for the inclusion-exclusion criteria, they may change, but we will always look at these three categories: muscle endpoints, things outside of the muscle, extramuscular, and quality of life. Because at the end of the day, it doesn't matter if you walk a second faster. If it doesn't actually improve your quality of life, it doesn't matter. And what we really want to learn is what's important to you. Now, so one of the questions we had is, will this improve my breathing? So I can't answer that. I can't say that. All I can say is that we're going to look for it. There's no reason to believe, based on the animal data, that it would or would not. We think it might have an effect because it does get into the diaphragm, but I can't tell if it's going to affect breathing, but it is certainly something we're going to check, and there is a chance it could. Is it going to affect the cognition, the thinking, the memory? We don't know. We know it gets into brain in animals, so we're going to check that. Somebody asked, one of the questions we got was about gastrointestinal function. That's a tough one, and that's one that I'm thinking a lot about, and I'm going to ask when we have our patient advisory boards about that, because I know that can be, I've heard that that's very troublesome to patients, and it's difficult to know how to measure that, how to assess that, because it's so different from patient to patient. But it's certainly something I'm very interested in looking at. And if we go to the next slide. Oh, before we go to the next slide, actually, so let me take a look at some of the questions that we've had, because I think I've tried to cover a lot of them. But how do I become the patient participant in the ATX-01 trial? Well, that will be, oh, here we go. Fantastic. How do I become a patient in the ATX-01 trial? Well, we will keep checking with the MDF website. That's the best place to check. And check with your doctor, because we're going to try to do a very big campaign to let people know when the study starts and to get them to the sites. There are sites scattered all over the US and Europe. Gut transit issues, this again is something I mentioned that we're looking on at. Anything for daytime sleepiness. I suggest you speak to your doctor. I can't give you any, although I am a neurologist, I can't give anybody any personal medical advice. All I can say is speak to your doctor, because there may be treatments that may be useful for you. Now, a cure for DM1. Well, cure is a big word, but we're certainly working towards that. But I think at this point in time, we're trying to do something. And the thing that we're doing that I hope has come through is that we're not just treating the symptoms with this treatment with ATX-01. The goal is not just to treat the symptoms. Yes, that's an important part, but we also want to do something to modify the natural history of the disease. And whether it's with our compound or some of the other companies' compounds that you heard of, I think we're all working very, very hard to try to get this disease to slow it down or even reverse it. And that's really important. And the congenital DM1 is an interesting question, because we're starting with looking at adult DM1, but there's no reason to believe this could not work for other forms of DM1. Now, the really interesting thing is that we think our compound might also be useful, and we're certainly going to test it at some point in DM2. And the reason is because unlike some of the other drugs that are in development, we're not focusing exclusively and specifically on the DMPK gene, because with DM2, it's a different gene. What we're focusing on is the MBNL, and MBNL is a factor in DM2 as well as DM1. So our drug has, at least theoretically, the potential to help DM2. So that's something that we have in mind, and Beatriz and her team are doing cell studies and cell lines from patients with DM2. I think we've talked about advances. I mean, there's lots of other trials that are ongoing as well. So, and I think if you come to this series, you'll hear more about these. We've talked about the preliminary results. I think this question is, what is the efficacy of the treatment? So we don't know yet in humans. We've shown you the animal data, but we'll see. We're certainly keen. We certainly think there's enough there to warrant going into humans. How soon before any drug is developed for human use? Well, there are drugs being developed right now. The question is, when will they be available? And that's always a difficult thing to predict. So if this first study that we're doing, for instance, is positive, then we will go on to do at least one more study, possibly more. So by the time the drug is released on the market, we're looking at maybe 2030. And I know that sounds really depressing, but it's actually not that long when you think about how long it normally takes to develop a drug. Now, the other thing that we're also very keen on doing is what's called early access. So depending on the results we see in the phase 1, we will consider, obviously, if we think it's safe and it's and there's efficacy, and I know there's a very big need after the first study of making this available through an extended access program. I think this would be something that we would consider. Now, those programs differ depending on which country you're in and what's allowed and what's not allowed, but stay tuned for that. So it doesn't necessarily mean that you won't be able to, as a patient, access the drug before then. But in terms of it being available on the market, I would say probably 2030 or later. How close are you to getting approved medications? We've talked about that. Where are you in the development for cure? When will this be available in Canada? So that's a good question. Usually Canada follows on from the US, but it's hard to say. Could they be in clinical trials this year? We are not, but I know some other companies are. Will this therapy be for a specific age group at any stage of progression? And as I said, we are starting with adult DM1, but we will eventually go to other groups. Are there treatments in the pipeline I would be eligible for? We've talked about the inclusion-exclusion criteria. I can't comment on a single person, and the target date to start a clinical trial will be early 2023. Let me ask Beatriz some questions here. I think a lot of these we've covered. Very new development. My frontal cortex muscle is now badly affected. I'm not sure I understand that question. Apologies. We talked about that. How soon we talked about that? I'll let Beatriz answer this question. Can the mRNA technology of COVID-19 vaccines be useful for the development of drugs for DM? I think one thing that it's done is it's brought the development of drugs for COVID-19 has brought to the fore the importance of RNA. So I think there's a lot more excitement about it, but I'll let Beatriz answer that question.
Yeah, I think it's a very good question because, thanks to the COVID, now we are all well aware of what RNA can do. And the thing is that, well, it's not exactly the same technology, but the basis is similar. What they are giving is just an RNA that they want to alert the immune system to react against. In our case, we are also using an RNA, but the contrary, we are protecting it so the immune system does not react and it can have a biological function. But yes, the idea is that we are using RNA technology. It is not considered genetic therapy because in fact we are not altering the genetic components of the patients. So we are just modifying levels of expression. There was also another question about something similar, this one about CRISPR.
Yep.
And so I think this question was about the genetic therapies. Right now in myotonic dystrophy, there are some companies working with CRISPR technology. Although this is used in genetic therapy, in their case, what they are doing is to target the RNA, so they will also have a treatment and not a cure, because using CRISPR technology or whatever other type of genetic therapy to alter the RNA, the DNA, so the genetics of a human being, it's still far away from the clinics. So. But yes, all these technologies are advancing very, very fast, and we will hopefully have more results, very good drugs approved from them quite soon. But in the case of DM, as I mentioned, these are very similar to, or similar approaches to the ones that are using ASOs, which is also RNA technology.
Thanks, Beatriz. Going through, I see we're sort of over time, so I'm going to try to speed up here a little bit and to focus on some of these. Does the therapy need to go through human trials? Yes, it cannot be available without human trials, and I think this is for everyone's benefit and safety. We talked about the diaphragm. What will it cost? We don't know at this stage. Obviously, it's really important to us that we ensure that we produce a treatment that is accessible to patients, because there's no good having a treatment that sits on the shelves. So the hope is that the patient cost will be very low, and that will be through making sure that we provide enough data that payers want to pay for this, or payers see the value of it. So that is something that we have to work very carefully on. We talked about how far along we are. We talked about when we're starting clinical trials. This is a really interesting question, I thought. I have no idea from my 57 years on this earth that I had DM1 until 2 weeks ago. Is it possible it is not as rare as it seems? I think whoever wrote this is on to something. I do think it is more common than people realize. I think there are people out there who have forms, who have different things that they don't put together, or their doctor doesn't put together as actually being DM1. And that rather than treating 5 different diseases with 5 different types of drugs, could be dealt with an anti-DM1 drug. I think once we have DM1 drugs on the market that are effective and safe, we're going to start seeing a lot more screening for DM1, and I think we're going to see these numbers increase. That's just my belief. When therapy is available, who and how and who will administer. So this is an intravenous therapy, and so it will be administered, at least initially, in the clinic by the clinic staff. There is a possibility that there will be home administration at a later date. That's to be worked out, but it will be available through neurology clinics, and certainly that would be the doctors who would be administering it. This is what we've talked about this, and we've talked about brain function. The hope is that it will, because it gets into the brain, but that's why we're doing the clinical study, so we can certainly test that. Is there another page of questions before we move on to the other?
No, this is the last one.
Okay. And then I think there was a slide on our pipeline, Beatriz. If you want to, I'll hand back over to you, and you can talk about some of the exciting things we're doing after this program.
Yeah, well, yes, we just to finish that, we believe that we could go beyond DM1. As you did mention already, the mechanism of action of our drug will allow treatment also for myotonic dystrophy type 2, and this is a real big unmet medical need, because there are no other developments that could target these patients that could be helpful for them. In addition, the reduction of MYR23B has been well studied in osteoarthritis, specifically in knee osteoarthritis. So what we are doing is also to start obtaining proof of concept of using anti-MYR23B by intra-articular injection here. And we have just started a new development in cancer cachexia with a different anti-MYR that hopefully will also be very helpful to cancer patients. And just to finish, I wanted to show you, last but not least, the team, the people that is doing this possible and that is working every day, yes, to bring these products into the clinic. So just to thank you very much for all your time. I think we did it too long, but I think it's worthy that we have time to answer all your questions. And well, just feel free to contact us at any time if you have more questions or if you want to know more about Arthex.
Beatriz, Dr. Llamusi, Dr. Walker, thank you both so much for sharing about Arthex, your company, and your products. I have learned so much, and I feel that we have all today learned a lot and gained more in our hope for cures and treatments for myotonic dystrophy. Thank you for your work and for being with us today. To our audience, I would say thank you as well. If you missed anything during the course of the presentation, this is recorded. You will be able to review this presentation online at the Myotonic Dystrophy Foundation website. And we will be very happy to see you at our next Meet the DM Drug Developer, Ask the Expert conference and other activities in the future. Thank you all so much, and have a great day and a great weekend.
Thank you all. Have a nice weekend.
Thank you.
Bye.
Thanks for your attention, and thank you to the MDF.